Hi everyone, welcome back to Useful Genetics. This is lecture 10M. We're going to continue our discussion of mobile DNA by talking about the RNA-based mobile elements, retrotransposons, the pseudogenes that these elements also create, and the extent to which all of this replicated DNA in our genomes can be considered to be junk. So retrotransposons are in many ways similar to the DNA-based transposons that we just discussed, but you see there's one big difference. So again, we have an element with repeated sequences, a particular recognition sequence at its ends, and an internal segment that codes for a protein. This segment is transcribed by RNA polymerase into messenger RNA, and it's translated into a protein. But rather than being a DNA-based transposase enzyme, this protein is an enzyme called reverse transcriptase. And it's called that because what it does is, in effect, it reverses what transcription has done, what RNA polymerase did. It takes the RNA and it makes a double-stranded DNA copy of it. Now, reverse transcriptase can act on any sequence that has at its 3' any RNA that has at its 3' end a copy of its recognition signal. And so, in particular, it does this on the messenger RNA from which the reverse transcriptase was synthesized on the ribosomes. So, reverse transcriptase makes a DNA copy of the RNA for the retrotransposon and inserts that retrotransposon into the chromosome or another chromosome at a new location. Again, it's replicated the element. Now, one distinction is that this replication is able itself to be replicated again because what's copied includes the promoter of the gene. So the promoter, the messenger RNA, starts upstream of the promoter. Unlike the promoters we've seen in the past, where the messenger RNA starts downstream, in retrotransposons, the messenger RNA starts upstream of the promoter, so the new copy also includes a promoter, is able to transcribe the gene for the reverse transcriptase, and is able to replicate independently of the copy that produced it. Now, like DNA-based transposable elements, retrotransposons can also act on defective copies of themselves. So the full-length functional copies of retrotransposons are mostly called line elements, where the L stands for long interspersed nuclear elements. But there are also many more copies in the genome of short versions of these elements, and these are shorter because they lack much of the open reading frame, and they're unable to produce a functional reverse transcriptase. But that doesn't matter because, as long as there's at least one line in the genome, because the sign elements still have a promoter, they can still make an RNA, and that RNA has the recognition sequence at its 3' end, so that the reverse transcriptase that's coded for by line elements can act on signs as well, inserting copies of them into the genome. And it can act on other elements, in particular the elements called ALU elements, the short 300 base pair elements that we looked at. They also have at one end, at their downstream end, a copy of the reverse transcriptase recognition sequence. And so reverse transcriptase will also act on them. And in each case, the DNA version that's inserted, the messenger RNA contains the promoter. So the DNA version that's inserted in the chromosome contains a functional promoter and is able to produce more RNA, which can then be copied into DNA again by reverse transcriptase. So these processes, as long as there are some functional elements that encode reverse transcriptase, these shorter elements are able to replicate abundantly in the genome. In fact, the shorter they are, it seems, the more effectively they replicate, and the more copies we have of them. Now, the third kind of element I want to talk about are elements called pseudogenes. And there's two categories of pseudogenes. You can guess from the name that a pseudogene is a sequence that looks superficially like it's a gene, but when you look closer, you realize it's a non-functional sequence that's homologous to a functional gene. And pseudogenes arise in two ways. One is they can arise by duplication of existing genes and then mutations that destroy the function of the gene. So they may have internal stop codons or frameshift mutations. They may have lost their promoters, but they still have the structure of a normal gene. If they have a promoter, they may even be transcribed and their introns may be spliced out. Often they're close to the functional gene because of the copying process by which they arose, the gene duplication process. However, the other kind of pseudogenes that I want to talk about, which are actually much more common, are called processed pseudogenes. And these arise as a side effect of the presence of reverse transcriptase in our nuclei. So reverse transcriptase, encoded by a line element or an LTRN element, another kind of functional reverse retrotransposon, can make a DNA copy of any messenger RNA that has a recognition sequence. And just by chance, many messenger RNAs will sometimes be acted on by reverse transcriptase, even though they don't have a very good recognition sequence, or sometimes no recognition sequence at all. Here's what happens. So any gene can be transcribed into RNA and processed by the splicing machinery into mature messenger RNA. And normally this mature messenger RNA goes off to the ribosome, to the ribosomes in the cytoplasm to be translated into protein. But sometimes, instead, this mature messenger RNA encounters a reverse transcriptase that uses it as a substrate. And that reverse transcriptase makes a DNA copy of the mature messenger RNA, double-stranded DNA, and then inserts that into the chromosome or a different chromosome. Now what we have in the chromosome is a copy not of the whole gene, but of the messenger RNA. So the introns have already been spliced out, and it has no promoter because conventional genes have their promoters outside of the start of transcription. So these sequences are now inert. Unlike ALU elements and short interspersed nuclear elements, they can't be further replicated by reverse transcriptase because they don't have a promoter. Now, let's back up and think about the effect of mobile elements on genome evolution in general. And you've had clues about this already. Basically, they're filling up our genomes. And this occurs over millions and millions, perhaps billions of years since the origin of eukaryotes. Mobile elements are also present in bacteria, but only in very small numbers. They don't fill up the genomes in the way that they have in eukaryotes. Even the introns in our genes, it's quite clear now that they began as mobile elements that inserted themselves into what were previously intact coding sequences. So our genome gets bigger and bigger and bigger as more and more elements insert, and each element in turn is also a site where more elements can insert. So it gets bigger and bigger and bigger. Should we consider that all of this sequence that's accumulated because of unselected processes of both the biases in DNA polymerases and the accumulation of mobile DNA, should we conclude that all of this stuff is junk? Some people want to argue that most of it is functional, but most molecular biologists who've given serious thought to evolutionary processes would say that, well, it's true that occasionally we find non-coding sequences that affect phenotype. And it's true that, some fraction, small percent of our genome is affecting phenotype. The latest data in a paper that just came out says maybe a total of 8% of the DNA in our genome is actually affecting our phenotype. It's under natural selection is how they detected it. But often people extrapolate from finding that, for instance, one sequence in an intron affects the expression of the gene to extrapolating and saying, well, all sequences in the genome, in the introns probably affect gene expression, when there's no justification for that. Or they'll use a definition of functional that's excessively broad. That means at some time, in some circumstance, it can interact with some DNA binding protein. In general, I think this is a circumstance where we really should be applying Occam's razor and choosing the simplest explanation that's consistent with what we observe. And in most cases, the simplest explanation is that the mobile DNA processes that we know about are sufficient to explain the accumulation of this DNA in our genomes. What are the consequences of all this DNA for the functioning of our genome? Well, the first consequence is that, of course, that as we saw in the graph, our genome is way bigger than it would otherwise be, at least 20, 30 times bigger than needed just to code for our genes. And this is not just more DNA that has to be replicated, more nucleotides that are needed, and more DNA replication machinery. It also, I think, poses a large burden in terms of the need to keep the DNA free of damage. Most of the DNA, having no function, is free to mutate. That's not as big a problem. But every time, even when the junk DNA gets damaged, it still has to be repaired because any damage to this DNA can prevent replication of the chromosome and thus prevent the cell from being able to divide. There are also mutational consequences of this DNA. Insertions, many loss of function mutations in our genes are actually due to insertions of mobile elements. These elements don't preferentially insert in unimportant places. They insert at random, and then natural selection has to deal with whatever harm is done. They also can cause changes in gene regulation. Because these elements themselves contain promoters, their promoters can drive genes expression in new ways. We saw this in Module 3 when we looked at how blood oranges came to synthesize so much red pigment. Elements cause genome instability. All of our discussion of chromosome rearrangements was processes that were almost entirely caused by the abundance of repeated sequences in our genomes. Incompatibilities between mobile DNA elements in different species or in different populations can promote speciation by making them unable to interbreed. And finally, it's certainly true that our genomes contain lots of randomly evolving DNA, and some of this DNA is going to turn out to be useful for later evolution. So we've considered in this lecture retrotransposons and pseudogenes, both simple pseudogenes and the process pseudogenes that are also generated like retrotransposons by reverse transcriptase. In the previous lecture, I complained about the sloppy state of our genomes compared to bacterial genomes, and I think we can blame it on the replicative copying and insertion of DNA transposons and retrotransposons and of all the passive sequences that they also act on. Coming up next, we're going to think about chromosome evolution. I hope to see you there.